Today we have the pleasure of having Iria Gomez join us, and she is going to present a talk titled Physiological and Pathological T-cell Autoreactivity Converge in T1D, an unexpected TCR story. And very interesting story indeed. So we're really looking forward to this. A short bio for her is that she's the principal investigator at the CIMAS Research Center and the associate professor at the University of Santiago de Compostela in Spain. Her main research interests are understanding the molecular mechanisms driving autoimmunity and developing novel drugs for the treatment of autoimmune diseases. She's been the recipient of 2 prestigious Marie Curie fellowships, and she's recently received the Career Development Award from Breakthrough T1D. I've dropped her recent paper in the chat. You can see it, and it's entitled Physiological and Pathogenic T-cell Autoreactivity Converge in Type 1 Diabetes. Thanks so much for joining us. We're excited to hear your talk.
Thank you very much for the invite and for having me here today. So I hope you can see the slides well. I'll be using the laser pointed. So basically, this is a story. It's a joint venture from the Pigment Bonifacio and my own lab. So this story started when we published this paper a few years ago in TCR beta chains as well in T1D. So for those of you that may not have seen it, basically what we did is we sorted true naive central memory Treg and TSCM cells from healthy donors and patients with T1D. We named them true naive because with this panel, we made sure we sorted separately the stem cell-like memory T cells, which is a subset that is usually within the naive pool. And we wanted to make sure that our naive cells were actually naive cells. We got around 200 million TCR beta sequences, and all of them are available at the iReceptor database. So please go ahead and play with sequences. And briefly explain this paper because it has some foundations for the one we published a couple of weeks ago. So basically what we found here, so I have something here that doesn't allow me to see the text. Yep. What we found is that TCR beta sequences show lower clonality or greater diversity in patients, which might mean that they are able to see more antigens than healthy people. And we also observed this displacement towards the left in the length of CDR3 beta chains. As you can see here in the gray boxes compared to the white boxes in all the 4 cell subsets. We also look at the sharing, the similarities in the repertoire in healthy people and in T1D patients, and we saw that among T1D patients, there is more sharing with each other, and it's important that we match for HLA as well. You can see here darker colors compared to the brighter colors here as well. So to understand what was happening with the shortening in the TCR beta repertoire, we went and looking at the pre-selection TCR beta rearrangement events. So for that, we did a bit of a trick, which is basically taking the out-of-frame sequences of your true naive cells. These represent TCR beta sequences that were generated in the thymus, but they were not subjected to thymus selection pressure. And as you can see here with the arrow, we also saw this displacement towards the left, indicating that T1D sequences were shorter as well. And when we look at T1D enriched and AT enriched clonotypes, these are clonotypes that only appear in patients or only appear in healthy donors. They also show these defective or altered rearrangement events. We also observe an enrichment in hydrophobic amino acids in a couple of positions, 6 and 7 of the CDR3. We also stand with a previous publication where we also collaborated with, where it was shown that in the Tregs and autoreactive TCRs, you tend to have more hydrophobic positions in that region of the CDR3. We then went a step forward and we asked the question, what happened with autoantigen-specific TCR beta sequences? Because as I showed you before, this was a study on the whole TCR beta repertoire. We found that GAD-specific TCR was also shorter than the average, And then we collated different TCRs from different studies on T1D, and in all cases, those sequences were shorter as well. So it seems that being short may be a feature of autoantigen-specific sequences. So we ended up with this feeling that there was obviously something different in the TCR beta repertoire, and that the antigen-specific sequences seem to have some features different in patients and healthy donors. So that's where the second story and the current story started. And as Monica said, this was published a couple of weeks ago when it's open access, and all the sequences are also available at iReceptor if you want to go there, download, and do your analysis as well. So basically, what we did, and we had also healthy donors and T1D patients, and from the same blood sample, we designed an experimental pipeline where we did several experiments with exactly the same blood draw. That would mean that we could compare different techniques, making sure that all the cells came from the same sample. We did ELISpots, activation marker-induced assays, and then with single cell sorted these cells through the index sort feature of the sort, we did single cell qPCR, then we sequenced these PCRs. And as we did our previous paper in the very same blood sample, we were able to track these antigen-specific sequences back in these deep sequencing TCR beta repertoires. We decided to do this with whole gut to be completely agnostic about the peptide epitopes that were relevant in disease versus health. We wanted to see how the responses were against gut, regardless of the epitope. And just to explain you who these cells are, so we did this AIM assay, the CD154C69 assay for CD4 cells. We single cell index sorted and performed single cell qPCR. This is not an X. We did by fluidine with qPCR specific for a panel of immune genes. And then we also sequenced the TCR. This index sort feature, for those of you which may not be familiar with it, allows you to sort the cells in different wells, and then you can go back to your flow data and see exactly where that cell appeared in other plots. For example, these are the plots for the true naive central memory, direct TSCM, and we can see whether cell in A1 position was a Treg, and whether the one in B12 was central memory, and so on. This allows for the post-sort analysis of surface phenotypes. So what we found? Well, we found the unexpected, right? We were expecting to find some enrichment by ELISpot or AIM assay for gut-specific CD4 T cells, but we found that they exist in similar proportions in healthy people and in T1D. I'm showing here ELISpot results for almost 100 donors. Healthy donors on your left in blue, T1Ds on your right in red. And as you can see, they behave similarly, interferon gamma and 10. I would even say that maybe the healthy donors do have more interferon gamma responses, and this was unexpected. When you look at the quality of these responses, as you can see, they are pretty much the same, interferon gamma alone, IL-10, or both. So they show us that ELISpot is great for some antigens, for example, peptides from proinsulin, but maybe not so for vodka. So we went into doing this AIM assay, and again, we found the same at the start disappointing result, where we saw that the frequency of these gut-specific cells was exactly the same in patients and healthy donors. And we move into looking at the phenotype, the show, the flow. And as you can see, many of them were of true naive phenotype, and around 40 to 50% of central memory. In one of the supplementary figures of the paper, we have the same for STV, the super antigen, and there were not that many naive cells. So it seems to be a feature of gut-specific responses. So after these initial disappointments with so many patients using gut and seeing absolutely nothing different, we move into doing the qPCR for the single cells. So we did the UMAP, you can see here the clusters, and they were very well defined. So what we found is, for example, cluster 9 is your typical Th2, and it has been shown that gut triggers an IL-13 response. We have, for example, this is Th1, Th17, some DFH-like cells around. These seem to be Treg-like cells, so they express, some of them express FOXP3 or CMAF, which is transcription factor related with posterior IL-10 secretion. And we have this strange Helios positive cluster as well, which was FOXP3 negative. 1 and 2 are more hyperresponsive energetic clusters. We look at the cell surface phenotype, and many of these hyperresponsive, they were indeed in green true naive cells. And then, as you can see, we have a balance of effector memory in orange and central memory in pink, showing that we have a mixture of memory and naive responses. So we were excited to look at, okay, where do the healthy donor cells and the T1D cells lie here? So red patients, healthy donors blue, they are completely overlapped. So we couldn't see a cluster that is specific of disease, but instead we found that it's not about the quantity of the cells, but instead of the quality. For example, there were some clusters where T1D cells did not express CTLA4, so that was suggesting a lower immunoregulation of these phenotypes, or, for example, increased expression of other genes such as CCR5 or EGR2. We look into the TCRs of these gut-specific cells. They were quite private, but we found, for example, when the same cells have the same alpha and beta chain, they were quite frequently shared with other clusters, including cluster 5, which was a regulatory one, suggesting that the same cell can trigger different phenotypes. And here we did glyph analysis. So then we moved to the, I would say, most powerful part of the paper. So basically, we took all these gut-specific TCR beta chains and tracked it back into our deep sequencing pools to see whether we can find them, how frequent they are, which are their features, and to see whether we could find a T1D specific feature, which was the main goal of the study. So we did this at 2 levels, nucleotide sequences and amino acid sequences. So why nucleotide sequences? Well, we postulated that it would be difficult to find the same nucleotide sequence in a different donor compared to the one it was originally generated, right? Because recombination is supposed to be random, and therefore the chances of that happening should be low. So we took these gut sequences, and the average frequency of tracking back was around 20%, plus minus 13, depending on the donor. And what we found is this staircase shape. What does this mean? For example, when we have gut-specific nucleotide sequence, if we find in the central memory subsets, they are found in the same donor they were originally generated from. These 3 phenotypes, for example, came from donor 1, and they were found in donor 1, and the same for donor 2, 3, 4, 5, 6. So this tracking back was ultra-private. And the same applied to Treg and TSTM. We have less dots here because, as you know, there are less Treg cells in the periphery, and therefore the depth of sequencing was also lower. However, we went into looking at the nucleotide sequences in the true naive cells. We found this completely different picture, and this is where the unexpected starts. So we found that these gut-specific nucleotide sequences are partly listed, in fact, in true naive cell subsets. We indeed took a couple of them, made transductions, and they are indeed gut-specific. So this will mean that our thymus allows gut-specific phenotypes to live in the thymus. Whether they are expanded or not, it depends on the person, but it seems that publicity is needed for gut. We also did the same for cytomegalovirus or CMV sequences in less donors, and we could not see this publicity, even though the tracking back was higher. We also look at the frequencies, right, of these nucleotide sequences, how frequent they are, because maybe they are really rare. Maybe they are found in central memory cells. It's got specific clonotypes in healthy donors, but they are so rare, they're not relevant. However, we did not find that. We can look here. So basically, y-axis is the frequency in blood. of these gut-specific phenotypes derived from healthy donors, from T1D, or CMV ones. And as you can see, the frequencies are pretty much the same. So we can see that gut-specific phenotypes expanded in the central memory pools are as frequent as CMV ones. And this is quite striking. This means that we needed those cells, and they got expanded, and we have memory. However, in Treg, although we had less instances, you can see that T1D patients seem to try to expand some phenotypes, and possibly these Treg phenotypes are not efficient in controlling the peripheral responses. So as I said, this was the first level at the nucleotide sequences, and then we moved into the amino acid level. So what is this? Well, maybe we have phenotype C, A, S, S, F, whatever, and this phenotype can be generated by several convergent nucleotide sequences, maybe 2, maybe 10, maybe 100. So we look at this and we found that publicity increases. As you can see, we have many different phenotypes, which are here in horizontal lines, and they are found in different donors. So publicity for gut is high. I don't have here the slide for CMV, but it was slightly lower, suggesting that we need a gut response. We also looked at the relationship between convergence and frequency. So the question here is whether are these phenotypes frequent just because they are easy to be generated? So this is what happens, right? What I'm showing here is the frequency of these amino acid phenotypes in blood. And here, how many nucleotide sequences converge into that very same phenotype. So correlation here is almost close to 1. In two naive cells, if a phenotype is frequent, it is because you have too many convergent sequences. It's easy to be generated and makes sense. But as you can see, there is a difference between the blue squares and the red dots. Red dots are T1D derived phenotypes. So what you can see is there is a huge number, I would say, of very frequent phenotypes that are also highly convergent. In summary, convergence is higher in T1D. And this is not that much for CMV. When we look at central memory, these phenotypes went into activation in the periphery, and then they came into the memory pool. I want to see how the convergence is there. Is it kept or is it not? One will guess that it's not. And indeed, that is the case for the blue gut-specific TCRs and healthies, where the slope is reduced, and the same for CMV. However, you can see the red dots, T1D phenotypes, are still highly convergent and highly frequent. So we don't know yet what is the reason, but it seems that highly convergent phenotypes are more easily expandable in T1D. We then look at the frequencies as we did in the nucleotide level, right? Are these gut-specific phenotypes frequent, rare? And we were expecting, or what we wanted to find is what is special in T1D, right? Which phenotypes are biomarkers, appear in T1D, they are not in HD. But we actually found that it's the other way around. What we found, what we did, let me drive you through this. What we did is we take gut-specific healthy donor phenotypes and try to find it out in the healthy donor naive cells. And this is the median frequency. And the same for gut-specific healthy in patients, and then the other way around. What we found is that the T1D gut-specific are highly frequent in healthy donors, basically the same frequency as if they were to be found in patients. So basically, T1D gut phenotypes are quite public, regardless of disease status. However, gut-specific phenotypes found in healthy people are rare in patients. Or if you say it in different words, this means that T1D patients seem to be lacking some healthy donor physiological, you call it TCR sequences. When we do the same exercise in central memory cells, we found that the healthy donor gut phenotypes are really frequent in healthies, and the T1D ones are also highly frequent in T1Ds in the same proportion. So healthy people do have an intense gut-specific memory. And this effect I showed you before, that rare in T1D, is also kept, as you can see here and there. We should look at the Treg repertoire also. Sorry, autumn is coming. the Treg repertoire, and as you can see, healthy donor-derived gut-specific genotypes are more frequent in Tregs in healthy, while the other frequencies are really low, including C and Bs. Finally, we went into wondering then what is special about healthy donor exclusive gut phenotypes versus T1D exclusive T1D gut phenotypes. What is the difference? What is going on there? So we found some differences in the V gene, J gene. These are not really important, really decisive, I would say. Some differences in amino acid usage. You can go and look at the supplementary figures. One thing I wanted to point out is more about where are they expanded in the UMAP space. So these are in blue HD exclusive phenotypes and in red T1D exclusive phenotypes. So if they are found in ternary cells, how they are phenotypically speaking? Well, they are interspersed pretty much everywhere, I would say. They are also indicate, sorry, phenotypes are expanded. However, when you look at central memory, this is more colorful. So we found more phenotypes in the central memory pool, makes sense. We found that gut memory is important. But we see that, as you can see, blue dots and red dots are not in the same space. So basically, blue dots, gut exclusive healthy donor phenotypes appear mainly in cluster 5 and 6, more towards the right. This is the Th1 and Treg clusters, so they seem to be related one to the other. While T1D is red, as you can see in the bar part here, they're more towards the left, this energetic or hyper-responsive, naive TGF-beta responses. And one striking finding is that we saw that those clonotypes that are expanded, they're only expanded in healthy donors. So these HD exclusive clonotypes are expanded in healthy donors. They're present in the regulatory cluster and they are missing in T1D. Therefore, we proposed this model where in healthy donors, the thymus allows for the selection of gut-specific phenotypes. So autoreactivity is allowed to leave the thymus. And we have these gut-specific ternary cells that may include these protective physiological clonotypes. So after birth, it's known that pancreas goes through neonatal remodeling, which implies physiological beta cell death and replenishment with new beta cells. This releases gut and therefore triggers the activation of physiological autoreactivity, which eventually leads to a normal repair and a functional pancreas. This process generates this gut-specific effector and memory cell that we've seen, this Th1, Th2s, and also this Treg. And this combines with other peripheral tolerance mechanisms that do work in healthy people. However, in T1D, it seems that this thymic selection allows for, or doesn't allow for, the generation of these protective clonotypes, physiological clonotypes. So when remodeling starts, there is this altered physiological autoreactivity that somehow leads to the incomplete repair. Memory generated in the same size, that's why we put exactly the same cell number here, but they have altered phenotype in the number, in the sense of gene expression profiles. And also the Tregs, they have different TCRs, they don't seem to be able to control this. And this is basically what we call diabetogenic autoreactivity, which obviously combines with failure as well to proinsulin and other autoantigens. And this combination leads to this massive beta cell death with the time, with years, and to avert T1D. And with that, I would like to thank you for being out there listening to the presentation, and thanks for Sugar Science for inviting me, and I'm happy to take any questions.
Fantastic. Thank you so much. I want to open it up to other questions. Anyone in the audience? You'd like to either unmute yourself, raise your hand. Oh, here's one from Remy Crusoe, Columbia. I may have missed it, but were all the donors HLA matched and of a specific HLA haplotype?
So what we did is we tried to match for in healthy donors and T1Ds for the same frequency of DR3s and DR4. So at least that was not a big issue to compare. And same age and sex as well.
Hi, Iria. Nice presentation and paper. Did you check whether test whether any of your GAD cells from the AIM assay did indeed recognize GAD?
So what we did is we did a cell line trolling for the gate, for GAD, and for CMV as well. So the cell lines, we generated cell lines for, I think, a couple of healthy donors and one patient, or the other way around, and they were GAD specific. And we then were asked by the reviewers to do some transductions from this public TCR that we suggested from two naive cells. So we did that for one healthy donor, one patient, and they were also GAD specific. Whether or not all of them are, that's a different issue, because we are working with the beta cell tracking back. It would be great to have it there. Shame is not there yet. But they are GAD specific, at least the ones we tested.
Thank you.
Great. Here's another question from Rita Lahemsa. Is the raw data available to be analyzed?
It is indeed. So the sequences are available, and well, they are in Geo and all those databases. They are not that user friendly. So we also uploaded them in iReceptor. There are two different studies. So we have the 2017 with all the deep sequencing to naive intramemory direct TSCM 16 healthy patients. And then we also uploaded the GAD specific and CMV ones with metadata associated as well. So age, HLA, and so on as well. So definitely it's open for people to use and reuse.
Yeah, I would also just note that Brian Corey's on the call, and he's the he runs the iReceptor, and we also have 1 billion TCR sequences on there currently that are specific to type 1 diabetes. So it's kind of a nice place to look at Iria Gomez-Touriño's work, as well as, you know, other scientists' data there, and compare contrast as you see fit. Thanks, Brian, for dropping the link into the chat. Remy asks, are there enough replicates to perform comparisons between HLA, DR4, and DR3 donors?
I would say that it depends on the study. Let's say, for example, for the spot, we have a decent sample size, like 50 versus 50 or 47. We did that, we didn't see any differences. Regarding tracking back, the sample size is lower, so I wouldn't be confident in comparing. I guess it will be maybe important to see studies in maybe DR3 only, or DR4 only, or in children. We did this in young adults. So we don't know whether this is a feature that is exaggerated as well in children that are closer to this neonatal remodeling. So definitely it was something worth doing.
Great. I just wanted to sort of like run through this question. It's sort of a bigger picture question. But, you know, given what you're showing here, can you comment on how these, you know, findings may inform staging in type 1 diabetes, as it's appearing with, at least with the GAD marker right here, that the disease is present sort of at birth in these endotypes?
Yeah, I guess this informs that we should be a bit aware that maybe GAD responses, if they are also present in healthy people, we need to have another control to check. We want to say that, yes, GAD is doing this, but maybe it's a feature of the age or a feature of endotypes, but not a feature of GAD. We were not expecting to find this. I mean, there were other studies suggesting that we all have autoreactive specific cells against GAD. We're not expecting this publicity and this really high frequency of the TCR beta chain. So what if we go into a trial with GAD and we see no results? Maybe it's because we are interfering with the physiological response instead of with the diabetogenic one. So it might be worth digging a bit more to see, obviously, the different epitopes. We can transduce this GAD specific healthy T1D and see what the differences are. But with regards of staging, I guess it's more about giving us more, opening new doors of questions that maybe GAD is important, it's definitely there, but we need to know that healthy people also have these memory responses.
Right, you have to be able to compare to the healthy population, I guess. What is the situation, or what is the possibility of looking for the shorter TCRs in the blood of a newborn? Is that even a possibility? is it the blood is, volumes are too big or no?
Well, it all depends on the depth you want to go. I guess then now the assays are better and they can be done with lower amounts of blood. But again, I guess it will be an expensive thing to do. I don't think we are quite there yet. It will be great to do the sequencing there. Possibly our antibodies are easier and cheaper and more worldwide accepted. But definitely this short feature, this high diversity in the TCR beta repertoire, this lack of physiological responses against, at least against GAD, it will be great to do it with proinsulin, with Z98, with other autoantigens, or even with vital antigens as well. We were surprised with the differences with CMV, that they have sometimes the same frequency. It's a response against an autoantigen as frequent as against a vital antigen. So it's definitely open new ways of thinking about autoreactivity, I would say.
So what's the next set of experiments for your laboratory?
I mean, we would like to possibly clone and study these healthy specific or healthy classic TCR sequences to see how they behave compared to T1Ds. And we, together with Mark Pigman and Ania Lorenz, we also have this HLA paper that's still to be finished up to give shape into these homozygous for DR4, homozygous for DR3, and heterozygous, where we did TCR repertoire as well to see what the difference are in the repertoires and whether we can find something different in heterozygous to tell us why they have this highest risk.
It sounds very good. Hats off to you guys for making your paper open access and for providing your data to an open access T1D TCR a data repo. So thank you very much for doing all the work and also then making it available to the scientific community. That's just, it's really laudable. Okay, if there's any more questions, last call for questions. All right, then. We're just finishing up here, and thank you again for joining us, Iria. It was really a great talk and an excellent paper, and we look forward to what your team finds next. Thank you again. Have a great rest of your evening in Spain.
Yes, so thank you for the invite, and thanks HiReceptor for the help in uploading all these sequences, which always takes some time. So thanks, Brian and Monica.
Okay, thank you so much. Bye-bye now.
Bye.